Okay, let me share my nervous system notes here. Okay, so we are beginning the nervous system. And just like the muscle system and the skeletal system, we're going to talk a lot about some of the organization of the nervous system, the different tissues. I will say that there's a lot to the nervous system. So take it in chunks. There are a lot of questions from this first chapter as well. Okay, and where I can, I try to give you hints, about questions and what I would ask, because there's a lot of within this first chapter, groupings of information, like the types of neurons and what are the neuroglia? Well, the two in the peripheral nervous system versus the four in the central nervous system. Those make great, easy questions. So I'll give you a heads up as we pass through these. So characteristics of the nervous system, they communicate with electrical signals. They're very rapid, specific, and produce an immediate response. Compared to the endocrine system, it's much faster acting, but both regulate and maintain homeostasis, and both use negative feedback and positive feedback loops, mostly negative feedback. Okay, so components of the nervous system. This is one of those pictures that you can come back and actually, if you have this printed off, is write notes on the side and embellish it with things so that one diagram kind of contains a lot of information. And this is one of those. So we have the central nervous system in tan in the center, which includes the brain and spinal cord. And then the blue and the pink regions are the peripheral nervous system. That's everything outside the brain and spinal cord. Right, and over here on the diagram of this woman here, the tan or gold in the center, the brain and spinal cord, is the central nervous system. And then the blue represents the sensory input, which are incoming signals generated from your organs, your hands and your feet that go to the brain and spinal cord. And on the left-hand side of the woman, we have the pink neurons there, and in the chart there to the left-hand side, the pink region, is the motor output. All right, these are signals generated by your central nervous system that travel away from the central nervous system, and stimulate, different organs, glands, or muscles, of the body. All right, so now it's showing sensory here on the left in blue and pink motor output on the right in pink there. But in reality, you have sensory and motor on the right side and on the left side. It's not just sensory is on the right input and motor output is on the left-hand side of the body. They just color-coded it that way. So in reality, pink and blue neurons are going to be on this woman's right side, and pink and blue neurons are going to be on her left side. All right. But when we look at this, we go, okay, if the sensory is incoming, and that's where the arrows are pointing to, right, all to the central nervous system, the sources are signals from the external environment, which you could even write in there right now, exteroreceptor, right? So the receptors that are stimulated by signals from the external environment are called exteroreceptors. So maybe you could write that next to that box. Then next we have signals from the skin, tendons, and muscles. Those are called proprioceptors or proprioreceptors. And then signals from internal organs. And next to it, you might write internal receptors. Right? So the receptors, from certain areas have certain names. And they convey signals either from the internal organs, skin, tendon, and muscles, or from the external environment. And that goes to the central nervous system. The response of the central nervous system is outgoing. So that's why you have the gold arrow pointing to motor output in the pink arrows or the pink neurons here. Right? It's either a single motor neuron to skeletal muscle, which is part of the somatic division, or a two neuron chain. There's one pink arrow, and then it splits into two arrows below autonomic nervous system square there to the parasympathetic and sympathetic. Right, so the autonomic nervous system is a two neuron chain, motor neuron, not just a single one, like in the somatic division. And the autonomic nervous system has two branches to it. Parasympathetic, which are your rest and digest physiological responses, and the sympathetic nervous system, which are your fight or flight responses. Right? Parasympathetic and sympathetic are in opposition to one another. So when you're resting, you do certain things physiologically, like slow down your heart rate. But when you're in sympathetic mode and you're in survival mode, then your heart rate increases. But we'll look at the autonomic nervous system, one of the last chapters. Okay, so parts of the nervous system, we have the central nervous system, composed of the brain and spinal cord, receives and processes and transfers information. Peripheral nervous system is everything outside the brain and spinal cord, and that includes your cranial nerves and your spinal nerves. I'll just tell you right now. Sensory neurons carry information to the central nervous system, and motor neurons carry information or signals away from the central nervous system. All right, so sensory input is information about changes in the environment that gets carried to the central nervous system. It's either like, we said before here, from in blue, external sources from skin, tendons, and muscles, or internal organ sources. Right. Integration sorts and processes and interprets the sensory input and makes an appropriate response. Integration occurs in the central nervous system, brain and spinal cord. And then motor output signals carried away from the central nervous system. And it's only so many things it can go to. It's either going to be to muscles or glands, right? To either contract these muscles, relax them, glands to either stimulate them to secrete something or to not secrete something. Okay, the division of the peripheral nervous system. Sensory division, signals carried to the central nervous system come from these three sources, right? Signals from the skin, tendon, and muscles. Proprioceptors, all right? So that's why I would go back to that picture. Signals from internal organs, internal receptors, and signals from the external environment, external receptors. So, that's why I'm often going to suggest just write those terms next to the blue box for that particular receptor. And that way you keep kind of copying it over and over again as well. The motor division of the peripheral nervous system is carrying signals away from the central nervous system. And we have two divisions. We have the single neuron that goes to this or participates in the somatic division, and that is the single neuron that goes to skeletal muscles, and it's very rapid. Or the outgoing signals are in this two neuron chain as part of the autonomic nervous system. Autonomic nervous system regulates smooth and cardiac muscle contraction and glands. The somatic nervous system is your fight or flight physiological responses. And parasympathetic nervous system are your resting and digest physiological responses. Okay, histology of nerve tissue. Well, we have two types of cells. We looked at this when it came to tissues, where we covered that, in exam 1 material. We had neurons and neuroglia. Neurons demonstrate electrical excitability and capable of conducting impulses, which means they carry these signals, or what's called action potentials. They respond to stimuli and convert to an impulse or an action potential. And so we're going to learn about these different action potentials, graded potentials, resting membrane potentials. But neurons are like electrical wiring. They carry a current, electrical current on them. Neuroglia are cells that support the neurons functioning in the central nervous system and the peripheral nervous system. Okay, so we're going to look at examples of these. Neuroglia, these supporting cells, they function to support, nourish, and protect. There are 6 types of neuroglia. 4 are found here in the central nervous system. 2 are found in the peripheral nervous system. Okay, so those found in the central nervous system, we have astrocytes. They form what we call the brain-blood barrier. The brain-blood barrier restricts movement of material that is, conveyed or part of the blood, like, found in plasma, and it keeps those materials in the blood vessel itself. And it protects the neurons and the different nerve structures, especially in the brain, because that's really important. So they wrap themselves around blood vessels and they discriminate or they disallow certain molecules or substances or hopefully organisms from ever going from the blood out into the tissues. Astrocytes, brain-blood barrier, that's going to come back later in AUP2. Actually, most of this comes back in AUP2. Oligodendrocytes are cells that are going to be similar to Schwann cells that we find in the peripheral nervous system, but these create a myelin sheath. And the myelin sheath is like insulation or plastic that you might find surrounding electrical wiring. So we insulate electrical wiring, so when a current passes along the wires, it doesn't just go out into the walls and become grounded, so to speak. Because that's actually what grounding is, right? With electricity in your house. So the myelin sheath wraps around it and insulates, parts of neurons called their axons, where these electrical impulses are traveling. And in doing so, it helps increase what's called the nerve conduction velocity, the speed in which this impulse is going to pass down the axon of one of these neurons. Okay, microglia, these are neuroglia that are capable of phagocytosis of different microbes and cellular debris, very similar to how white blood cells will work in AUP2. You're going to study that more there. Remember, phagocytosis is engulfing something and creating a vesicle around it. And then we have ependymal cells that produce and circulate cerebral spinal fluid. Right? Ependymal cells produce and circulate cerebral spinal fluid. All right, so these are just some pictures of, what they consider the different shapes of these cells. I think it's kind of interesting to see the astrocyte because that forms the brain-blood barrier. See how they're wrapping around these capillaries because capillaries have blood inside of them. And then we have microglia. They're capable of phagocytosis. We have the ependymal cells to the lower left-hand side that line, what's called these cavities, fluid-filled cavities of the brain called ventricles. And those cells are actually going to produce cerebral spinal fluid from capillaries that are nearby, and they're going to push this fluid out into this cavity and let it circulate around the brain and around the spinal cord and actually through the center of the spinal cord as well. We'll look at a couple pictures of that. And then the oligodendrocytes are the ones, oligodendrocytes, are the cells that wrap around the axons of these different neurons and with what's called a myelin sheath. And the myelin sheath, is kind of a fatty layer that insulates the wiring here so that the impulses will travel more rapidly down their axons. We'll look at, we have multiple videos, in the notes for the nervous system because there's a lot to it. The nervous system is like the skeletal system and the muscular systems combined, plus a little bit more. So there are plenty of videos for this. This is, again, one of those PowerPoints of notes that I would save to thumb drives. I would save all my notes to a thumb drive, even if you have a habit of going to the course page and studying from there, and opening up the notes, is I would keep a copy of everything because it's going to be very helpful in AUP2. Okay, the neuroglia in the peripheral nervous system includes Schwann cells, which, like the oligodendrocytes in the central nervous system, produce a myelin sheath. And that helps insulate the axon and it helps increase what's called the nerve conduction velocity. And then we have satellite cells that are found around cell bodies, and they help regulate the exchange of material between the cell body and the interstitial fluid. They also help to cover certain regions so that not all neurons can interact with the next neuron, maybe in a chain. It'll cover up maybe some binding sites or receptor sites. Unmyelinated axons or cell bodies appear as gray matter. So as we look at models of the spinal cord and neurons in the peripheral nervous system and then spinal cord and brain in the central nervous system. You see a lot of gray stained structures and regions, and then I guess you would call it like a vanilla color, like a tan color regions or whitish kind of colored regions. And where we have gray staining, darker staining tissue, it means we have cell bodies or axon terminals or parts of an axon that do not have any myelin. Because the myelin is going to be kind of fatty. So where we see fat, we think yellow often on models or white. So where we have white matter, it will be representative of axons of neurons that have myelin sheaths. If it's gray matter, like the outer 2 millimeters of your cerebral cortex is your gray matter, that's where we have cell bodies or unmyelinated structures. We have gray regions in the spinal cord as well. Kind of looks like a butterfly shape. And so this will make a little more sense when we see that peripheral nervous system and central nervous system models. Okay, so this also shows you where we would find in the peripheral nervous system satellite cells, which surround the body and regulate the passage of material into and out of the cell body across that plasma membrane, and regulates, and can kind of cover receptor sites on the cell body or on dendrites. Then we also have the Schwann cells, right? And they wrap around axons. So the axon is going to be the nerve fiber they're pointing to here in yellow. The Schwann cells wrap around and around and around and around and around, kind of like, if you had, for girls or well, for guys too, I guess, that might take their hair and roll it up in a curler. If you're going to curl your hair, if you have long hair and it was straight, roll it up in a curler. That to me, what a Schwann cell looks like. It wraps around and around and around and around, and it covers a certain region of this nerve fiber. And then there'll be another Schwann cell next to it that will wrap around and around and around and around as well. You'll have these little regions of nerve fiber that don't have any myelin sheath, because the myelin is going to be in the light blue here. Those are called the nodes of Ranvier. So the myelin sheath doesn't cover the entire surface of that nerve fiber, but the most of it. Okay, and this shows you what the Schwann cell, how it begins. It makes contact, especially embryologically, and soon after birth, these Schwann cells go to town, covering, the axons of the nerves in the peripheral nervous system. And so it starts on the surface of the axon, an A, or actually a number 1, because it says a Schwann cell envelops an axon, and it wraps around and around and around and around, so that you finally have multiple layers of this neurolemma, that insulates that axon. The nucleus of the Schwann cell is the pink there. Okay, so parts of neurons. Neurons are the cells that conduct impulses. And we have 3 different structural types. We have multipolar neurons, bipolar neurons, and unipolar neurons. And they're going to be found in specific areas. But they generally have the same parts to them. And then we will look at, the difference between these 3 different types. They all can conduct impulses. And we will see, the similar parts of them, structure, dendrite, cell body, axon, and so forth. This will coordinate well with your lab class, hopefully. I'm not exactly sure about the online standardized lab classes. I actually have yet to teach one of those. I've taught the AMP2 lab for a long time, what we call like the standardized eCampus, version. But then, I teach them face-to-face, and I have some of my face-to-face versions that are online, but I've never done the AMP1. So I'm not exactly sure. I'm pretty certain that it's very similar to how they do AMP2, where they have a mixture of different models and pictures from the standardized lab book that they use, as well as some that are models that are used throughout virtually every campus, of the college. But this will still help you quite a bit, even if it's slightly different. Okay, the dendrites. I'll get back to work here. The dendrites are processes or extensions on the cell body that have receptors on their surfaces. And they're capable of responding to changes in the environment, because that's what all receptors do, right? All right, so they, once stimulated, can conduct an impulse called a graded potential toward the cell body. Right, so it's sort of like branches on a tree. You have these branches that extend way outward, that might have leaves on them, but then you have like the core and the center of the tree for which the branches all grow away from. That's the dendrites are like the branches. Okay, then the cell body, also known as the soma, contains the nucleus and the nucleolus, surrounded by cytoplasm and typical organelles that you would see, in any other cell. Then we have the axon, and between the cell body and the axon is what's called the initial segment, right? So I'm going to come back to that. But we have the cell body. The axon are nerve fibers arising from the cell body that are capable of conducting these impulses called action potentials, all right? Where the cell body meets the axon is this kind of funnel-looking, and I don't mean funny, like a funnel, or a triangular area, called the initial segment or the axon hillock. Right? And it's a cone-shaped elevated area where we generate or we initiate these action potentials or impulses or signals. Right? So we're essentially going to take the information that's gathered from the dendrites, from the branches, on this tree. And they're going to send signals through the cell body down to the initial segment. And if the signals are significant enough to send, an action potential down the axon, then it will do so and initiate it here at the initial segment. So it's a place where you kind of decide, do I send a signal or not? And you might not. So you never send an impulse down the axon. But if it's significant enough, we generate what's called an action potential. And the axon is like a current traveling all the way down the axon to the axon terminals where we release acetylcholine or a neurotransmitter. We looked at that, in the muscle physiology videos, where we talked about how muscle contraction starts with an impulse traveling down the motor neuron. And as it arrives at the axon terminals, calcium, voltage gated calcium gates open and calcium enters the axon terminal, and that stimulates the vesicles to merge with the membrane and release the neurotransmitter acetylcholine. All right. So this is what we're talking about. Now we're kind of looking at, well, what's happening along that neuron? All I jumped in on was, hey, it talks about this signal traveling down a motor neuron. And now you're learning what this motor neuron or these different neurons are. All right. So, the dendrites are these specific little processes. The cell body contains your nucleus and nucleolus. We have an initial segment that kind of joins the cell body to the axon. And then axon travels whatever distance it needs to. Some axons are very, very short. Some are very, very long, and maybe travel all the way from your lumbar spine to the bottom of your foot. That's how long they can be. Okay, nodes of Ranvier. When we looked at this picture up here, and I said that the Schwann cells are here in blue, and the nerve fiber in gold there that doesn't have any myelin sheath and light blue covering it, that little naked region there, that's called a node of Ranvier. Okay, so the node of Ranvier is an area on the axon where an action potential can occur and where there is no myelin sheath. All right, so it's going to be these little gapped areas between myelin sheaths, what I call a naked axon or nerve fiber. And action potentials are going to be recreated in each of these little naked regions. And again, we will have a video that's going to clearly demonstrate that. Okay, the axon terminals are the distal end, the dilated terminal end of a neuron, where we store those synaptic vesicles, and synaptic vesicles Remember, a vesicle just stores something. So synaptic vesicles are storing neurotransmitters in a neuron. And a neurotransmitter is a chemical signal released from a neuron that will bind to a receptor of another cell. And so it's like the neuromuscular junction from the nerve to the sarcolemma. In the nervous system, we'll have often, instead of nerve to muscle, we have nerve to nerve to nerve. And those are called synapses. But we still communicate the same way. You release a neurotransmitter, and then it needs to bind to a receptor on the next cell. Okay, so here we have a picture of a motor neuron, and it has this typical shape to it. You have a big cell body here with extensions called the dendrites, right? So they kind of look like tree branches. There's receptors, different types of receptors on the surface, the generally ligand receptors. We have a nucleus in the center. You can see some typical endoplasmic reticulum, mitochondria scattered there. And then you get to this axon hillock. So it kind of looks like a funnel, right? Or a triangular kind of area here. The axon hillock, or the initial segment, is where these different signals from the dendrites will be assessed, so to speak. And if we need to send a signal, an action potential, down the axon, all the way down to the lower right-hand corner to the axon terminals to release acetylcholine, I shouldn't say acetylcholine, a neurotransmitter, because it may be a different neurotransmitter. Then we generate that impulse here at the axon hillock, or the initial segments, as often referred to. Right, then we have in the center here of this whole motor neuron, in the center in gold, is the axon, or the nerve fiber. It terminates to the lower right-hand side as the axon terminals. In there, we have the vesicles storing neurotransmitter. In blue are individual Schwann cells that have wrapped around the axon. All right, so wrapped around. Each of those little areas of blue represents one Schwann cell that's wrapped around. And the little gap of axon in between two Schwann cells is a node of rhombium. It's a node of rhombium. Okay, so in lab, we have these models, which are really good for identifying the different parts of a motor neuron. Right? And because, I'm going to put it this way, it would be a very long, large model if we had the whole cell body and the axon, and then a long axon with Schwann cells all wrapped around it, forming the myelin sheath, in one linear orientation. So they have the cell body and part of the axon here at E. And then below that, see where to the far right-hand side of E, you can see where it's kind of cut there. Then they take that part of the axon and they put it below. So you can't quite see it right now, but you can just way below D here, see the Schwann cell. Here's the nucleus of a Schwann cell down here. So we'll see more of that axon that has myelination to it below. But we took this picture with one of the cameras and just took this part first, and then the next one will have the lower part, if that makes sense. If you get a chance, come on to campus so you can see these models. All right, so here, this whole large region here is the nucleus, or the soma, or the cell body, or the soma of this motor neuron. And the nucleus is C, right in the center. Okay. These gray branches, so to speak, that are extending off the cell body are your dendrites. All right, that's what D is pointing to. This red region here between the cell body and E, which is the axon, that red triangular region kind of looks like a funnel. That is your initial segment, or your axon hillock. A, they look like little brown chocolate kisses. Those represent the axon terminals of other neurons in the environment. All right, so we're going to look at this one motor neuron, but these they look like little chocolate kisses here. Those are the axon terminals of other neurons. And in the base of that, like A was pointing to one of those, in the dilated region of A are going to be synaptic vesicles storing neurotransmitter. So when we look at this, I call it the chocolate kiss model. We'll come back and maybe look at this model again so that you'll understand what A is trying to identify for you. Okay, then we look at the lower part of this model, and we have in the center the axon, right, which is what C is pointing to, right? They cut it open to the left-hand side of C so you can see all the mitochondria and different organelles found within the axon. Okay, so the axon is right through the center. Right? Wrapping around and around and around is the Schwann cell, and the nucleus of the Schwann cell is D here, that pink. All right. The white layers here that B is pointing to is the myelin sheath that this Schwann cell has wrapped around and around and around the axon. So you can see these multiple layers to it. So that is the myelin sheath. Where the myelin sheath of one cell meets the myelin sheath of another cell is here at A, the node of Ranvier. So it looks like these myelin sheaths are actually touching one another, but they're not. So they don't exactly touch, and they don't exactly cover the entire surface of this axon. There's going to be a portion of it that is actually naked, even though you can't really see it on this model. We know it from this diagram here. They're trying to represent this node of Ranvier. So although it looks like these myelin sheaths touch, there's actually this microscopic area of the axon that does not have any myelin, and therefore this electric conduction can actually occur there. Okay, and so, and then lastly, E is the connective tissue layer that surrounds one neuron, and that would be the endoneurium. The connective tissues surrounding nerves and fascicles and individual neurons are the same as what we looked at in the muscle. We had an epimysium, paramysium, and endomysium. In the nervous system, we have an epineurium around the entire nerve, a perineurium around a fascicle, and an endoneurium around one cell. Okay, so cluster and bundle terminology. All right, we have, both in the peripheral and the central nervous systems, we have clusters of cell bodies and we have bundles of axons that are going to travel together. But the terms are different in the peripheral nervous system versus the central nervous system. So it's helpful to understand the difference between them. So clusters of cell bodies in the peripheral nervous system are called ganglion. Clusters of cell bodies in the central nervous system are referred to as nuclei. Bundles of axons in the peripheral nervous system is just called a nerve, but bundles in the central nervous system are called tracts. Okay, so structural diversity, motor neurons, or neurons, I should say, can have three different basic shapes, and therefore they are also going to have certain functions associated with them. We have multipolar neurons, which is what the shape of the model we just looked at. We call it the Lisa Simpson model because it kind of looks like it's either Lisa Simpson or her younger sister that had the braids that's, that kind of stick up from her head. So that's what it's always reminded us of. So we have multipolar neurons, bipolar neurons, and unipolar neurons. Multipolar neurons are motor neurons. And the interneurons, we'll look at these association or interneurons within the spinal cord itself. They're the same shape also, right? So multipolar neurons or motor neurons, the neurons that are conveying action potentials or signals away from the central nervous system. I'm going to quick page up here. Hold on. Back to the beginning, where we had that body. Okay, here. Pink to the right-hand side. The motor output, either the single motor neuron, somatic, two motor neuron, autonomic nervous system. Above motor, I would write multipolar neuron there because all those motors are all this same shape. They're multipolar neuron shape. And while we're here on the sensory side in blue, you know, because this is coming from every place in the body except for your special senses. Next to that sensory input box there, I would write unipolar neuron, because all these sensory neurons that are conveying signals from internal organs, the muscles, you know, from the exteroreceptors, the proprioceptors, interoreceptors, they're all of the same shape. They are unipolar neurons. However, sensory information from your special senses, like vision. So maybe I would draw an arrow from the eye of this woman standing here to the central nervous system, to the brain spinal cord, from the nose to the brain spinal cord, maybe from the ears to the brain spinal cord, because those are all special senses, right? Smell, vision, and hearing. Right? Those neurons, those particular sensory neurons, are going to be bipolar neurons. So while we're here, sensory from eyes, nose, and ears to the central nervous system, just draw an arrow there, and then write bipolar neuron. Over the multimotor neuron output, the motor output here, I write multipolar neuron. And over sensory there in blue, I would write unipolar neuron. While we're there. Okay, so let's look at these. Multipolar neurons carry motor, these are the motor neurons and interneuronal association neurons. We'll see these in some models coming up, especially when we get into the peripheral and central nervous systems. Right, bipolar neurons, as I mentioned before, are sensory neurons from eyes, ears, nose. They convey sensory information from special senses. Almost all the special senses are going to use this bipolar neuron. And then conveying somatic sensory neurons, which are the neurons from the sensory neurons that are conveying sensation from the three blue areas we just saw, from the interoreceptors, the proprioceptors, the exteroreceptors. They're all of the unipolar shape. Right? So that's what we call somatic, because it's general body sensation. Touch and stretch, you know, they convey sensations like touch and stretch. These unipolar neurons have kind of a hanger shape. I'll show you in just a minute. To us, they look like a hanger. All right. So the multipolars over to the left-hand side. All right. This is similar to the model we looked at, where we had a cell body with dendrites extending from it. I mean, these are pretty long dendrites, but you get what I mean. And then you have an axon leading away, and it's going to terminate in the axon terminals and so forth. Okay, that they convey, this is the most abundant type in the body, and it's the major neuron type. You know, it conveys motor information. So all the motor neurons are multipolar in their shape. All right, special senses. All right, these are bipolar and rare. They're found in some special sensory organs like that convey olfaction, which is smell, you know, vision and hearing. They're going to be of this shape. So look, this is a different shape. It's very linear. We have dendrites right to a cell body and then an axon. It kind of looks like tree branches on both ends. All right, but very linear. Unipolars are found mainly in the peripheral nervous system, commonly only in the dorsal root ganglion, which means that they are conveying sensory information from, you know, general bodies, about general body sensation. They have a different shape. So we have these peripheral processes extending from the axon here like a tree branch. But look at the cell body, and then this it's kind of like a neck region between the cell body and the axon here. You know, at the other end of this axon, you know, they have here the central process of the axon, and then it extends down below. They both kind of look like tree branches on both sides. But, you know, we kind of think that this kind of looks like a hanger, like you could hang like a coat on each side of these axons here, and then the cell body is kind of like the hook that would wrap around. Well, anyways, kind of looks like a hanger. So unipolars, different shape than a bipolar, different shape than a multipolar. Okay, the synapse. The synapse is where two neurons meet, where one is conveying maybe an impulse, like a neuromuscular junction, where the nerve and the muscle meet. Well, this is where a nerve and then another nerve in a sequence of cells communicating with one another are going to meet. Nerve impulses pass from neuron to neuron at synapses, moving from the presynaptic neuron, which is the neuron conducting an impulse, the first one, to a postsynaptic neuron, the neuron that you're going to now communicate with. So here over in the upper right-hand side, it shows you an axon, kind of like what you looked at the neuromuscular junction, an axon ending in an axon terminal here. Right? And then you have the next neuron. Here are the dendrites. They usually terminate on dendrites, and sometimes even the cell body is showing you like that on the one on the lower left-hand side is another neuron trying to talk to this multipolar neuron, but it's actually going to make contact or release its neurotransmitters to be able to bind to receptors on the cell body, not necessarily the dendrite. But each location, dendrites and cell bodies, may have more or fewer receptors to different neurotransmitters in the environment. Okay, so in the at 12 o'clock up here in gold, it's showing you this synapse, where you have a presynaptic neuron, a postsynaptic neuron. We have models in lab that show this as well. So, here we have the axon, at about 2 o'clock here on this image in gold, right? And it terminates on the axon terminal here. You can see these little synaptic vesicles within the axon terminal. When it's stimulated, you can see these little blue dots are being released into this gap area between one neuron and the other. And it's similar to the neuromuscular junction, where neurotransmitter is released, it crosses that gelatinous gap, and then can bind to a receptor on the next cell. So this is a rather helpful picture. Okay, electrical signals in neurons involves ion channels and membrane potentials, resting, graded, and action potentials. All right, so we're going to look at what these different channels are that allow different ions to pass through. Different channels respond to different stimuli to open or close. All right, so that's why they have different needs. And then the potentials, we have three different kinds: resting potential, graded potential, and action potential. And we'll talk about what a potential is. Okay, so first we have to start with the ion channels. We have leakage channels, which just randomly open and close, and they leak ions across the membrane. Now let's go back and quick review. The ion, or the cation, that's in highest concentration inside cells is always going to be potassium. So it's high concentration inside and low concentration outside. And the cation, positively charged ion, that's in highest concentration outside a cell is sodium. So sodium is in high concentration outside the cell and low concentration inside the cell. And we said what maintains that gradient? The sodium-potassium pump. 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. That is there, abundantly and works very hard in these neurons and in muscle cells. And part of that, they work like that is because we have leaky gates or channels that randomly open and close. And some of these leakage gates are for potassium and some are for sodium. You actually have more potassiums than you do sodiums, but anyway, we'll get to that. All right, so some of these just randomly open and close. And so that's another reason why you need to have these sodium potassium pumps to put these ions back. Okay, so that's a leakage, randomly open and close. Then we have voltage gates or channels. They open upon the change in the membrane potential, right? So when a certain charge is acquired across the membrane, then certain gates are designed to open up. Some open up at negative 55 millivolts. Some open up at negative 50 millivolts. They generally, sodium and potassium are going to open at the same time. When the same voltage is acquired, one opens and closes more quickly than the other, but they pretty much open at the same when the same voltage is obtained, and it's about negative 55 millivolts. So when a certain charge is acquired, these channels open up. Then we have ligand gates, and they open due to attachment of a hormone or maybe a neurotransmitter. So when a chemical compound binds to it, that stimulates to open. So it doesn't matter what the voltage is across the membrane at that point, is if acetylcholine or norepinephrine or, whatever it might be, or insulin, which is a hormone, binds to a receptor, boom, it's going to open up. It's not dependent on the voltage. And then we have mechanical channels that open due to vibration, pressure, or stretching. Vibration, pressure, or stretching. All right, those are 4. Those you should know. All right, the distribution of ions, as I mentioned before, potassium ions are the major intracellular positive cation, which means it's a low concentration outside the cell, and we already see a diffusional pathway. Potassium, if it could, would go from in the cell to out of the cell. Sodium ions are the major extracellular positive cation. So now we know what direction sodium wants to move. It's high concentration outside the cell. So if one of those channels or gates open up, sodium will enter the cell. Right, but right now, potassium high concentration inside the cell, sodium high concentration outside the cell. This distribution is largely maintained by the sodium-potassium pump. This pump actively transports 3 sodiums out of the cell and 2 potassiums in. 3 sodium out, 2 potassium in. It is active, which means it requires ATP. Okay, love this picture because we're going to come back to this again. Where it color codes a motor neuron here. All right, so this is a multipolar. This is a motor neuron, or a multipolar neuron, where we have dendrites at one end, then we have the cell body, then we have the axon here in green, and we have the axon terminals, in red. So they look like, dilated fingertips of these little hands here. Okay. All right. Color-coded are the types of gates we expect to find in these regions. So on the dendrites in the cell body, we expect to find blue. And that is blue because we have chemically gated channels there, or ligand. Chemical and ligand are the same thing. I need to update that, I guess, and put chemical there with ligand, because the textbook uses ligand, and then they started using chemical soon after. But ligand and chemical are the same thing. Okay, so on the dendrites and cell bodies, we have chemical gates there. So a hormone or a neurotransmitter needs to bind to it. Then along the axon here in green and branching down to the axon terminals, the green area represents the presence of voltage-gated channels. And these voltage gates are generally for, or almost exclusively for, sodium and potassium. So along the green area, we have voltage gates for sodium and potassium there. And then at the little dilated regions here at the axon terminals, we have voltage gates, but for calcium. Now, this is part of what that other video in the neuromuscular junction was talking about. They said an action potential travels down this motor neuron. And when it arrives at the axon terminals, and it says in there a couple of voltage-gated, calcium channels open. Calcium enters the axon terminal, and that stimulates the synaptic vesicles to merge with the membrane and release the neurotransmitter. Right, so we're kind of coming back to that and reinforcing that again. Right, voltage-gated calcium channels are found on the axon terminals. Along the axons, all in green here, are voltage gates for sodium and potassium. And on the cell body and the dendrites, we have blue areas. Those are chemically gated or ligand-gated channels for neurotransmitters or hormones. Now, we're also going to come back here, and while we're here, I'm going to mention it, is if you could, and you printed this out, is to the side here where you have all the blue area, the dendrites and the cell body, This is what we call graded potential territory. This is where graded potentials are generated and conducted. Right? So the blue area is also going to be a graded potential area. The green area, right, the green area on the axon and leading down to the axon terminals, where we have those voltage gates, that is action potential territory. Action potential territory. Right? So while we're here, it's just kind of helpful to write that in. Blue is graded potential territory. Green here is action potential territory. And we're going to talk about what those are right now. There are three types of potentials: a resting membrane potential, action potential, and graded potential. All right, so a resting membrane potential is the charge across the membrane of the cell when it's at rest. It's not conducting an impulse. It's not being stimulated at the dendrites. It's doing nothing. It's like having the light, turned off, in your living room, or the television turned off. It's not doing anything. All right. Now, when you want to turn it on, you're going to flip a switch, right? And then a current is going to travel to it, and the light bulb comes on, or the television screen illuminates, and a series of reactions occur. But when it's off, it's off. Right? And that's what we refer to as a resting membrane potential. If we can then measure the charge across the membrane to the immediate inside of the membrane compared to the outside of the membrane of the cell, the inside is always rather negative, a negative value of about negative 70 millivolts. All right, so we're going to show you a picture of, if you had a voltage meter and you could measure, one end to the inside of a cell and one to the immediate outside of the cell, on both sides of that membrane, then the inside would be negative compared to the outside. So 70 millivolt difference exists between the inside and the outside of the cell. Inside of the cell is negative relative to the outside. Thus, it's the resting membrane potential when it's not actively doing anything is negative 70 millivolts. Sodium potassium pump maintains this or restores this. Okay, so here is a picture of a little voltmeter. We have a probe to the inside of the cell, and then we're also measuring the outside of the cell. The inside of the cell is negative. That's why you have all those little negative marks there, and the outside is rather positive. In gray, that represents the plasma membrane. And so we're looking at a region, let's say on this axon, has a little box down there, and it's going, okay, if I can measure the inside to the outside, the voltmeter says in that little box up on the top, negative 70 millivolts. Now, when we look at these videos and they're showing you the changes in charges and ions moving, they almost always have a little voltmeter there. So you can see what effect the flow of sodium into the cell is having on the charge, across the membrane, or the potential. Because it's got to go from negative 70 to maybe negative 50, and maybe all the way up to plus 30. And then it'll become negative again. And that will be due to the movement of potassium ions, but that becomes another part of this lecture. So almost always they're going to have these videos where they're going to show you these different ions moving, and they'll have a little voltmeter up there so you can see what's happening to the charge across the membrane, to the potential. Eventually, we're going to graph this and we're going to label it. Okay, so the resting potential is just a little redundant to what we just said, but the average is negative 70 millivolts. The voltage difference between the inside and the outside of a living cell is the resting membrane potential, and is said to polarize the cell membrane because it's kind of like a polar covalent bond. One end is positive, one end is negative. Well, here, the inside of the cell is negative and the outside of the cell is positive, so it's called being polarized. Potential energy exists because of the different ion concentration gradients that exist. Remember, sodium is high concentration outside, so it wants to, diffuse into the cell where it's a low concentration, and vice versa for potassium. It's high concentration inside the cell. It wants to diffuse out of the cell. Okay, graded potentials versus action potentials. So graded potentials occur in certain places and has certain characteristics, and then action potentials as well. Right, so graded potentials occur on dendrites and even cell bodies. So that's why I said up here in this picture here, the blue area of the dendrite and cell bodies, this is where graded potentials occur or pass through. All right, oopsie doo. Right, so they are generated really here at the dendrites. They travel short distances to that axon hillock or initial segment, and they can vary in size and strength. Right, so we're going to see that because they might make or generate a very small difference in the membrane potential or a very large difference. And the difference may make the interior of the cell become more positive, or it may make the interior more negative. It just depends. I mean, there's variability here. But action potentials are different. Action potentials, there is no variability. They are generated or occur here on the axon hillock or the initial segment. They travel away from the cell body on the axons, right? And they are one size, one strength. The wave that they produce, when we graph it, the wave, or the difference in the membrane potential is always going to be the same. It's going to go from -70 ultimately to about +30, and then back to ultimately -70 again. Right? So one size, one strength. They can travel long distances because it depends how long the axon is. If it's a very small motor neuron, within the spinal cord itself, it doesn't have to travel very far. But like I said before, some motor neurons are going to travel from your lumbar spine all the way to muscles, in your calf area or in your feet, or from your neck into your, forearm and into your hands. Those are long distances, right? So they're going to be highly myelinated. They're going to have a lot of myelin sheath on them. And they are all or nothing or none principle. Once we meet the threshold point at the axon hillock, where we decide, do we send this signal or not on the axon? And once we reach the threshold point, which will be -50 millivolts, we send that signal no matter what. But if we don't reach the threshold and the potential is kind of bouncing around maybe -60, -58, -72. If you don't reach the threshold point, then you do not send the actual potential. It's like having a bunch of dominoes all lined up. And unless you push over that first domino, you're not going to knock down the rest of the dominoes unless someone slams the door or something. But you get what I mean? If you put enough force into that first domino to topple it over, then the rest of them are going to fall down. Okay, so resting membrane potential, graded potentials, and action potentials. This graph is excellent. If you can reproduce this and label this up in multiple ways, you are in excellent shape for understanding these different potentials, and it will help you when you get to the cardiac chapter in A&P2. All right, on the vertical axis, we have millivolts, the membrane potential in millivolts that we're measuring on that voltage meter. And then on the horizontal or x-axis, we have time elapsing. And so we start the clock at like 0, and then time elapses 20 milliseconds, 40, 60, 80, blah, blah, blah. All right, so let's look at the coloring here because this all makes sense. Here, let's start at the dotted line. That is threshold. All right, the dotted line is threshold, and that's at -50 millivolts over to the left-hand side. Below that in the yellow area means, we are below threshold. And then above the dotted line in the peach background there, that means we are in action potential territory. Okay, so we said that generally, at least for these types of tissues, your resting membrane potential when the cell is not at work is -70 millivolts. So we're going to start here at -70 on the left-hand side. And we start the clock running, and we here at rest, and that's the flat line there of that blue line, all right? And nothing's happening, the cell is at rest. These arrows here on the bottom represents some kind of a stimulus, something happening on those dendrites or cell body, right, that cause the interior of the cell to become less negative or more positive. Like here, after the first black arrow, you have this upward deflection, which means that when we're measuring the voltage across the membrane, whatever happened here on this dendrite or cell body caused the interior to go from about negative 70 to just about negative 60 millivolts. So it became less negative or more positive. That's called being a depolarizing gradient potential because it's taking us closer to zero, or what's called an excitatory postsynaptic potential. An excitatory postsynaptic potential, EPSP. We're going to get to that. All right, so that little upward deflection short of the dotted line means that this is a gradient potential, right? It caused the interior to become more positive, but there was just this one stimulus, and the sodium-potassium pump got everything back to where it should be. So we go back to negative 70 millivolts. All right, then we have this other stimulus, the next black arrow. That causes the interior of the cell to become more negative. Because look, it goes from negative 70 to about negative 77 millivolts. And then again, we go back to resting membrane potential because the sodium-potassium pump does its job. Right. When we become more negative than we started, it's called a hyperpolarizing gradient potential. Right? A hyperpolarizing gradient potential, or hyperpolarization. When we become more positive in the cell, it's called depolarizing it. When we become more negative or negative again, it's called hyperpolarizing. Okay, so that's arrow number one. Arrow number two, then we go past 60 milliseconds, and we have two arrows in a row. So what they're saying is that, hey, if you have two stimuli, that arrive very close together, that they will, what's called summate, they will add together like a tsunami, all these waves that join together in a tsunami. So we're having what's called wave summation. Right. So before, here we have one stimulus, the first black arrow, that causes the interior cell to become more positive. And before the sodium-potassium pump can put everything back, we have that other black arrow really close together to the first one, and that causes the interior to become more positive even more. Right? They add together or summate. Now, still, even those two do not get you to the dotted line. Right? So this just remains as a gradient potential that's being generated on the dendrites of the cell body when it arrives at the axon terminal or the axon helic. I mean, not the axon terminal, the axon helic or the initial segment. We are not generating an action potential on the axon. Sodium-potassium pump kicks in, and we go back to resting membrane potential at negative 70. All right, now we're at about 90 milliseconds. Now we have three black arrows back to back, boom, boom, boom. And the effects of them causing the interior of the cell to become more positive add up or summate. And look, now we hit that dotted line. Now at the axon, the initial segment, or the axon helic, that signals the neuron to convey an impulse or an action potential down the axon. And this upward deflection here, the upward spike of that blue line, and then the downward deflection of that blue line, that is the action potential. Action potentials will always have that same shape. And they really should have this going up to plus 30 millivolts because that's pretty much what it is all the time. I'm not sure why, because in the video, it talks about plus 30 millivolts. So I don't know why this image isn't, or this figure, this diagram isn't consistent with that. But there's some inconsistencies in Myriad. Anyway, so all the little Humpty Bumpties here that are short of the threshold point, the dotted line, those are all gradient potentials being generated from the dendrites or cell body. And when they arrive at the axon helic, they're not significant enough to throw the switch, so to speak, to the light and let the current travel down the axon of this neuron. But if it does get to about negative 50, negative 55 millivolts, whatever the threshold is for that cell, then it generates the action potential. And that's the big blue wave there that you see. The upward deflection portion of that is called depolarization. And we're going to look at exactly what gates are opening, what's closing, what ion is moving. then it peaks out, generally should be at plus 30 millivolts, not 10. And then we have the downward deflection, which is called repolarization. We actually overshoot our resting membrane potential of negative 70 millivolts. So that little overshoot region there is called hyperpolarization. And then we use the sodium potassium pump to get back to your resting membrane potential, which is your negative 70 millivolt flat line there. There's going to be one part in one of the videos where it says that the leakage gates help you get back to resting membrane potential. That is not accurate. They keep telling me that the people who make the videos are different than the, the writers and, of the textbook, because in the textbook, it clearly indicates that sodium potassium pump does that. So there's sometimes some little boo-boos here and there in these videos, but otherwise they really give you a good visual of what's happening here. And again, I would use these to kind of go over and over and watch them 3 or 4 times, because it's rather painless to do until you can practically recite the video yourself, and then you have it down. But here on this graph also, where you have these little upward deflections, these depolarizing graded potentials, while I'm here, I might put an EPSP next to that. And even where you have the 2 black arrows and the 3 black arrows in a row, these are all little graded potentials. And they're called excitatory postsynaptic potentials because it causes the interior of the cell to become more positive. Where you have that one hyperpolarizing graded potential, the second black arrow on the bottom there, I might write IPSP next to that, because an inhibitory postsynaptic potential causes the interior of the cell to become more negative. It causes hyperpolarization. So another term for this hyper-negative state, or looking at that, we can term that as an inhibitory postsynaptic potential. Okay, we'll get to those. All right, and we're about there now. Impulse generation. Right. So depolarization. We said that when the cell becomes more positive in that depolarizing graded potential, and then the upward deflection part of that action potential that should really go up to plus 30, right? That is called depolarization. A decrease in the membrane potential, which means, that you're becoming either less negative, because if you're negative at negative 70, decreasing the membrane potential means we're going to go from negative 70 to negative 60 or negative 50 or 0 or to plus 10. Right? You're going to become less negative or more positive. Right? So that's going to be upward deflections of those lines. Right? Hyperpolarization increases the membrane potential, which means in this case, you're going to go from negative 70 to even more negative, like negative 80 or 85. And when that happens, it gets you farther away from the dotted line or threshold point. And so it's called inhibiting that cell, because if the charge across that membrane is farther away from ever getting the threshold, then we're inhibiting that cell. If we're making changes or stimulating that cell to allow it to become more positive and approach threshold, then that's called exciting it, because it gets it closer to actually sending that excitatory impulse or signal on the axon. Threshold, at threshold, the membrane is depolarized to negative 55. Sometimes it's negative 50, but it's often one or the other, negative 50 or negative 55. You will never be given both of those options as a multiple choice, answer. Okay. So at a particular voltage, voltage-gated sodium and potassium channels begin to open, thus the beginning of an action potential. And that's what's important about the threshold, right? Is the threshold point is when the sodium and potassium gates become sensitized, those channels, and open, and allow sodium to move into the cell and potassium to move out of the cell. That creates the action potential. All or none, an action potential either happens completely or doesn't happen at all. So it's like turning on the light switch or turning on a light. When you flick the light switch, you either turn it on and the current moves, or you maybe toggle that switch a little bit, but if you don't quite, turn the switch already, to the on position, then you're not going to have a current traveling on the wires and illuminate the light bulb. Okay, graded potentials can be excitatory or inhibitory. So as I was describing before, the graded potentials in the blue area of that one picture, that dendrites and cell bodies can vary. You can have a neurotransmitter bind to one of those receptors and it causes the interior to become more positive, or another neurotransmitter or a hormone that binds to something, and it causes the interior to become more negative, and hyperpolarizing it. So we just have terms for that. EPSPs are excitatory postsynaptic potentials, and they cause depolarization, a positive deflection towards zero. So an upward deflection, becoming more positive or less negative, however you want to think of it. IPSPs are inhibitory postsynaptic potentials. They cause hyperpolarization. That's the downward, the negative deflection, on those diagrams. They get you farther away from threshold. What happens to the neurotransmitters? Really the same that happens at the neuromuscular junction. Those neurotransmitters either diffuse or they get broken down by an enzyme. Before we were looking at acetylcholinesterase. And then some of them that can actually be recycled. We even saw that where, the portions of acetylcholine were endocytotically taken back into that cell because it might as well recycle some of those parts that it can use, and sometimes, it'll even completely reuptake the entire neurotransmitter. It doesn't have to be necessarily enzymatically broken down. Okay, so they can diffuse, they can be degraded by enzymes, you can have reuptake, that should say bi cells. I'm sorry, that's a typo. Not B cells, it should say bi cells. Let me write that down because that's going to make me crazy because I see these little glitches and I forget after I'm done. Removal of neurotransmitters slide to adjust it. And then I see it again next semester and I'm like, why didn't I change that? So, okay, I just wrote that. Okay, so that takes us to, again, we were now taking these EPSPs and IPSPs, where now it's completely written out. On the top here, the top blue indicates the response to a stimulus, now in a pink arrow, an excitatory postsynaptic potential, an EPSP, right? That stimulus causes the interior of the cell to become more positive. Below that is an inhibitory postsynaptic potential. Again, with a pink arrow, causes the interior of the cell to become more negative. And then back to resting membrane potential. That's really all you need to know from that. You don't necessarily have to know, which ions are moving which way, but it makes sense, is that, and this will, I think once you do the action potentials and we look at those videos and we identify, hey, when we have the action potential and we're starting that at threshold, the upward deflection is called the polarization. Well, which voltage gates are opening first and which ions are moving first to cause that cell to become more positive, and then what happens to stop it at plus 30 millivolts, and now what's causing it to become more negative. So once you pass through that a couple times, if you want to come back and read this, it will make sense. The only way we can become, more positive in the cell, is to allow sodium to enter the cell somehow. Or if we could lose some kind of negatively charged ion, some kind of anion within the cell to leave. Because if we lose some negatively charged things, or if we acquire positively charged things to the interior of the cell, both of those are going to make the charge inside the cell to become more positive. And in the same respect with an IPSP, if we need to become, or we're going to become more negative in the cell, there's just two choices. We either lose positively charged ions like potassiums, or maybe we acquire negatively charged things like chloride ions, and that will cause the interior of the cell to become even more negative. But I generally won't ask you about those kind of things. What I will ask you about is the waves of, the different portions of an action potential in which ions are moving in which direction. And there's actually, I will tell you, is that there is a picture on the course page that I drew out by hand that is essentially something like this, where I'm writing EPSPs, IPSPs, and when it comes to the action potential part here, I'll say, okay, sodium gate opens, sodium enters the cell, or sodium enters that region, that upward deflection is called depolarization. And then at the top of the peak, it's going to say sodium gate closes. And then next to it, potassium gate opens or channel opens. That downward deflection is called repolarization. And it's going to say potassium leaves the cell. And then we have the downward deflection till it finally stops at the bottom, and that is where the potassium gate closes. Right. So at the bottom there, when we kind of bottom out, that's where I would write potassium gate closes. The overshoot, the portion that is from -70 to maybe -75, is the area of hyperpolarization. And then we go from the lowest point here back to your resting membrane potential of -70 millivolts. And we say this is we say restore resting membrane potential, and that is accomplished by the sodium potassium pump. 3 sodium out, 2 potassium in. So there's actually something handwritten on there. I'll try to share that with you in just a minute, like when I get to the end, and I'll show it to you. Okay, so this takes us to our first video. I'm going to go ahead and play this so that hopefully you can hear it. Remember, you can always put these PowerPoints in slideshow mode like this, and as long as you have internet, it should connect and run. So you just hover over down here where you see that arrow and click it.
The human brain alone contains about 100 billion nerve cells, called neurons. A neuron, like every other cell, has positively and negatively charged ions, inside and outside. Further, a resting neuron has a greater negative charge on the inside surface of the plasma membrane and a greater positive charge on the outside surface. This partitioning of charge creates a voltage difference across the membrane known as the resting membrane potential, which can be measured using a voltmeter. On average, an intracellular electrode records a value of -70 millivolts. The resting membrane potential depends on 2 factors. First, it depends on the presence of sodium and potassium gradients across the plasma membrane. Specifically, there are more sodium ions outside the neuron than inside, and more potassium ions inside the neuron than outside. Second, the resting membrane potential depends on the differential permeability of the plasma membrane to sodium and potassium ions. Leak channels in the plasma membrane allow sodium and potassium ions to diffuse or leak down their concentration gradients. The membrane contains many more potassium leak channels than sodium leak channels. Thus, the membrane is much more permeable, or leaky, to potassium ions. As positively charged potassium ions leak out of the neuron, the inside surface of the membrane becomes negatively charged compared to the outside surface. If potassium was the only ion moving, the potential would stabilize at -90 millivolts. However, positively charged sodium ions leak into the neuron, which slightly offsets the negative charge and raises the voltmeter reading to -70 millivolts. Sodium-potassium pumps actively transport sodium ions out of the neuron and potassium ions back in, compensating for the sodium and potassium leaks. Thus, the pumps help to maintain the resting membrane potential.
Okay, so yeah, I like these. Okay, so here in this diagram, they're showing you that, okay, here's a multipolar neuron. We're on a dendrite, right? And they're showing, this is a little redundant because this is, again, IPSPs versus EPSPs. But they're showing you again, hey, you know, if I had the voltage meter present here and I could measure the inside and the outside of the membrane with a depolarizing stimulus or an excitatory postsynaptic potential, an EPSP, it causes the interior of the cell to become more positive, and that's what it's responding to. Hyperpolarizing means we can become more negative again. You can kind of look at that on your own. I don't want to belabor this because I feel like I'm being a little too verbose here. I'm being a little too chatty. Maybe I should just read these notes, but hopefully you're getting the concept here. Okay, chemical synapses, which are most of what we're going to be looking at. Involves axon terminals on a presynaptic neuron, contains synaptic vesicles, which contain neurotransmitters. So you have 2 or 3 neurons in the chain. You start with the first neuron that's conducting an impulse, and it arrives at the axon terminals, like we saw in the neuromuscular junction video, and it's going to stimulate these synaptic vesicles to release their neurotransmitters. The receptors for the neurotransmitters are going to be present on the next neuron, or what's called the postsynaptic neuron, on the other side of the synapse. The cleft, the synaptic cleft or gap, is the fluid-filled space between the neurons. So, yeah, very similar to the neuromuscular junction. Conduction across the chemical synapse. So we have the action potential that travels down that first neuron, right? The presynaptic neuron. Calcium voltage gates open to, and allow calcium to enter into the axon terminal of the first neuron, or the presynaptic neuron. That triggers exocytosis. The synaptic vesicles fuse with the membrane and release the neurotransmitter. Neurotransmitter travels across the synaptic cleft or gap and binds to a receptor on the next neuron, or the postsynaptic membrane. Chemical or ligand-gated ion channels open in the postsynaptic membrane, because remember, the receptors are going to be on dendrites or a cell body. That's the blue area on that diagram back there. That's where we have the ligand or chemical gates found there, or channels found there. And depending on which channel opens as a consequence of acetylcholine or norepinephrine binding to it, it will cause the interior to become either positive or more negative, and thus create either an IPSP or an EPSP. Okay, so this takes us to the next video. Let's hope. Some of these run well, and sometimes I have to modify it. Yeah, this one, I'm going to have to check to see why it's not connecting. Maybe if I click here. No. It's supposed to have a link to it. It's not that great anyway. I wouldn't worry about it. But if I can, and I'll try to work on this, and then I'll re-upload these notes. So by the time, if you even listen to these Zooms, you have the notes that are accompanying it. I might have already fixed the problem, but right after I'm done with uploading the Zoom, I'm going to see if I can get this to work properly. My apologies. Right, generation of an action potential. So we looked at these graded potentials, dendrites and cell body, but now we're looking at, hey, what is the action potential? What is this signal that's generated in the axon hillock, or the initial segment, that travels down the axon? Right. So the resting membrane potential, or when it's at resting membrane potential, voltage-gated channels for sodium and potassium are closed, right? Because the cell is at rest. But once we, reach a certain threshold at that funnel area at the axon hillock, or the initial segment, if it reaches negative 55 millivolts or 50 millivolts, but generally it's negative 55, then the sodium gates rapidly open up, and sodium starts to move. Potassium gates will open, but they open much more slowly, and they close very slowly. Sodium gates open and close really, really quickly. Okay, so that produces what's called the depolarization, the upward deflection portion of the action potential wave that we're going to see drawn out, right? So the first part is depolarization. Graded potentials increase the permeability by opening sodium chemical gates, and sodium enters the cell. This is the beginning of depolarization. When depolarization reaches the critical level called threshold, right, sodium voltage gates open and sodium rushes into the cell, which begins the generation of an action potential at the axon hillock. All right. So what does this mean? This means that somehow, at the axon hillock, you had to have become more positive to reach negative 50 millivolts, because you were formerly negative 70. So sodium is entering when those ligand or chemical gates, are being opened. Right? And when that stimulus arrives at the axon hillock, then that's now on the axon in the green areas of that diagram we looked at earlier is action potential territory. And this is where we have voltage, not ligand, but voltage gates. So once you had that negative 55 millivolts, these gates start to fly open, like you're knocking over one domino to another, to another, to another. And the first ones to open up are the sodium ones. And in doing so, sodium, which is positively charged, rushes into the cell. And so now you're acquiring all these cations, these positive charges, and it causes the interior to become more positive, all the way up to about plus 30. Right? And that explains the upward deflection. When you're looking at these videos, try running it and just looking at the little voltage meter they're going to give you. And as they're talking about these things, you can see the charges, change. Okay, the action potential becomes self-generated, or a positive feedback mechanism. As sodium enters the neuron through voltage gates at one end, it causes sodium voltage gates next to it to open. So it's sort of like, I'm going to try to come up with an analogy, but I guess let's say you have a sink full of like a sink that has like an inch or two of cold water on the bottom, and you're like, gosh, I need to wash dishes, and it needs to be hotter than this. So you just put on the hot water all by itself. As the hot water drops into the cold water, it initially is warm right where the water is entering, at the end of the faucet, at the base of the faucet. But as it circulates, right, then the colder water to the periphery of your sink, eventually the water all starts to move, right? I mean, not just from the force of the water dropping down from a faucet into the water, because gravity is now stirring it up, but the warm water is going to diffuse into the cold, and the cold is going to diffuse through the warm. So eventually it just becomes all the same temperature. That's the best way I can describe it. As sodium enters in one area, and it's going to show you on the video, is that, it's reaching threshold, and it's met threshold, I should say. Sodium is rushing in, and it sort of floods from the channel that's initially allowing the sodium to pass through. It starts to travel down inside of the plasma membrane to the adjacent area. And that now takes that from negative 70 to negative 55 millivolts. And now that sodium gate opens. Now even more sodium is flooding in. That affects the resting membrane potential of the neighboring plasma membrane to go from negative 70 to negative 55. And now that sodium gate opens. So it happens all the way down the axon. So that's what they're referring to here. Okay, so depolarization, the upward deflection, is due to sodium gates opening and sodium entering the cell. Eventually, that sodium gate closes. And now finally, the potassium gate opens. So when the potassium gate opens, potassium is going to go from inside the cell to outside the cell and diffuse out. Now you're losing positively charged things, and it causes the interior to become more negative again. That explains the downward deflection of the line on that action potential. That's called repolarization. The potential moves back toward a negative value, and that's due to the potassium leaving. Before the passing gate actually closes, we hyperpolarize, we overshoot, we go past negative 70 down to about, negative 75. That brief overpolarization or hyperpolarization area, is concluded or stops when the potassium gate finally closes, and potassium is no longer leaving the cell. Now we go back to, or we have the period of restoring and going back to negative 70 millivolts, back to our resting membrane potential. And that is accomplished by the sodium-potassium pump. 3 sodium out, 2 potassium in. 3 sodium out, 2 potassium in. All right, so you have repolarization. What's opening? What's closing? Which ion is moving in what direction? And then that gate closes, and now potassium finally opens. That is repolarization. What direction is potassium moving? When does the potassium gate close? What is your area of hyperpolarization? And then how do we get back to resting membrane potential? How many ions in which direction? Okay, so this is like a brief diagram of that. One is indicating the resting membrane potential. We have graded potentials that are occurring, and at the axon hillock, we finally reach the threshold point, the dotted line, to generate the action potential. So the action potential part is actually that part of number 2 that is above the dotted line. That's called depolarization. I need to have a sip of coffee because I'm getting tired, I guess. And you see how depolarized we go all the way to plus 30 millivolts. So I don't know why the other diagram doesn't show plus 30. Anyway, depolarization. Sodium gate opens, sodium gate closes. Now at the peak at 30 millivolts, finally, the potassium gate opens. So the potassium gate opens. That causes 3, repolarization. We become more negative. Right? And at 4, finally, the potassium gate closes. Between negative 70 millivolts, where that number 1 kind of is, to number 4, that area is your hyperpolarization. Then we have restoration of the resting membrane potential back to number 1 on the right, and that is due to the sodium-potassium pump. 3 sodium out, 2 potassium in. This shows you the different types of stimuli that are causing these EPSPs and IPSPs, and how they can wave summate. So here up on the top, we have this one dendrite that has, it's called, excitatory number 1. It's supposed to be an excitatory postsynaptic potential, and this is the first example, or this is neuron number 1. One impulse travels down it, right? And here's E1 down on the bottom in the blue here. It goes, okay, that pink arrow, that's, this is when that impulse hits that dendrite there. And it causes the interior to become more positive. It doesn't reach threshold, the dotted line, but it causes it to be more positive. All right. Then your sodium-potassium pump gets you back to resting membrane potential. And then here they're going, look, here's another signal traveling down that neuron. And again, it has the same strength and amplitude, and it causes the interior to become more positive, but still never generates an action potential because you never get to threshold. Right? So it's called being, and it says on the bottom, 2 stimuli separated in time cause EPSPs that do not add together. So they're too far apart. The sodium-potassium pump can do its job, and they won't add together. But temporal, or this type of summation, it doesn't matter whether it's temporal or spatial. I just kind of want you to see the different ways that this can happen. On that same neuron, what if we have 2 impulses traveling down that neuron that are really close together? So down on the bottom in the blue box, you have E1, a pink arrow, and another pink arrow. And now they're so close together that they add together and they summate. And together, they're able to reach threshold and create the action potential. Or a different combination, you could have 2 different neurons, one communicating to one dendrite and one to another dendrite or a cell body. So they're calling it E1 and E2, excitatory neuron 1, excitatory neuron 2, whatever. And if they're both carrying impulses to release their neurotransmitter and they're excitatory in nature, they both cause the interior to become more positive, and they both arrive on those dendrites and the cell body and ultimately the axon hillock at the same time, then they too will add together and summate and produce an action potential. Now, the last example to the right-hand side is showing you two different neurons, but one is going to release a neurotransmitter that produces an excitatory postsynaptic potential. The other one in red, that's why they have I1. That's an inhibitory type of an impulse or effect of the neurotransmitter. So E1 makes the interior become more positive. The I1 causes the interior to become more negative. So on the bottom here in the blue chart, under D here, the first stimulus is saying, hey, if even if we don't have anything on the green neuron, excitatory one, if we have a stimulus coming, the stimulus coming down the inhibitory one causes the interior to become more negative. It causes it to hyperpolarize. All right. And now a little time elapses, and now an impulse both on the green excitatory and the red inhibitory neurons kind of arrive at the same time. Then what happens is they essentially cancel each other out. The negative one will be a little negative and the positive one will be positive, but they essentially cancel each other out. So there's different combinations that can occur. Okay, that takes us to the next video. Neurophysiology. This is what they call the generation of an action potential.
Neurons send signals over long distances by generating and propagating action potentials. Most action potentials originate near the axon hillock of the cell body in the initial segment of the axon. It then travels the entire length of the axon. A closer look reveals that during an action potential, voltage-gated channels open and close, altering the permeability of the plasma membrane to sodium and potassium ions. A threshold stimulus changes the shape of the voltage-gated sodium channels, causing their activation gates to open. This event marks the beginning of phase 1 of the action potential, known as depolarization. As sodium ions diffuse into the axon, the membrane potential becomes less negative. This causes more voltage-gated sodium channels to open, and the membrane potential soars to plus 30 millivolts. At this point, two key events occur. The inactivation gates of voltage-gated sodium channels close, and voltage-gated potassium channels open. These two events mark the beginning of phase 2 of the action potential, known as repolarization. As potassium ions diffuse out of the axon, the membrane potential becomes negative again. However, the membrane potential continues in the negative direction, going beyond the resting state of minus 70 millivolts. This marks the beginning of phase 3 of the action potential, known as hyperpolarization. During this phase, voltage-gated potassium channels close and all voltage-gated sodium channels are released from inactivation. By the end of this phase, ions move through leak channels only, and the membrane potential returns to the resting state of minus 70 millivolts. The neuron is now ready to fire another action potential. Summary: generation of an action potential. A threshold stimulus opens voltage-gated sodium channels. Sodium ions diffuse into the axon, depolarizing it to plus 30 millivolts. Voltage-gated sodium channels close, and voltage-gated potassium channels open. Potassium ions diffuse out of the axon, repolarizing it to a negative value. The membrane potential briefly hyperpolarizes. Voltage-gated potassium channels close, and the membrane returns to the resting state of minus 70 millivolts.
Okay, so propagation of impulses. The way we conduct these impulses, the action potentials, along the nerve, Right? So conduction of nerve impulses or action potentials. Right? So there's two different methods, and essentially you either have axons that have the myelin on it, or you have a naked axon with like no myelin or very, very little myelin on it. And the advantage to having these Schwann cells or the oligodendrocytes and this myelination is it allows these impulses to travel more quickly. So this diagram or this video is actually going to show that. Right? So continuous conduction is a very slow method of conduction, and this is the kind of conduction that's going to occur on unmyelinated axons. Right? So essentially, this depolarization, repolarization, hyperpolarization, opening and closing of sodium potassium gates, when we have continuous conduction and there's no myelin, is going to have to occur all the way down the length of that axon. And finally, when it gets to the axon terminals, then we can stimulate the voltage calcium gates to open, and that will stimulate, essentially the release of the neurotransmitter. But it's a very slow, tedious process. Saltatory conduction occurs when we have the myelin, right? The myelinated axons with those little naked regions called the nodes of Ranvier, because only at those naked little regions of the nodes of Ranvier does this process of the action potential have to occur, right? So it almost looks like you're skipping a stone across the water, on top of a smooth lake. It's going to travel much more quickly. There's other physics associated with it, but the bottom line is that the whole process of depolarization, repolarization, hyperpolarization, back to resting membrane potential, blah, blah, that is only going to have to occur at the nodes of Ranvier. And so it arrives at the axon terminal much faster, and you ultimately release your neurotransmitter more quickly or more rapidly. And that's what we call increasing the nerve conduction velocity, the velocity at which the action potential arrives at its destination from where it's generated at the initial segment. Okay, so propagation of action potential, similar to a domino effect, the voltage change in one area affects the adjacent parts of the membrane, similar to what we were talking about there in that video, whereas the voltage gate of sodium opens, sodium enters, and it starts to change the charge of the membrane adjacent to it and gets it to, or stimulates it to get to threshold. Okay, not all graded potentials become action potentials, as we saw. Sometimes you never reach the threshold point, right? During action potential generation and for a short time afterwards, the neuron cannot respond to another stimulus. This is called the refractory period. You have an absolute refractory period and a relative refractory period. But the absolute refractory period is based on the fact that once these voltage gates open and then they close, they kind of have to stay closed for a certain amount of time before they can open again. So what that ensures is that the stimulus doesn't start traveling away from the cell body toward the axon terminals and then somehow turn around and come back again. It can only travel away from and in the forward direction. So it ensures each action potential is separate, an all or none event, and enforces one-way transmission of the action potential. Okay, so this is one of the last videos.
Action potentials propagate in a continuous fashion in unmyelinated axons. Once an action potential is generated in the initial segment of the axon, it propagates the entire length of the axon. Recall that a threshold stimulus causes voltage-gated sodium channels to open. The influx of sodium ions generates an action potential. It also establishes a depolarizing current that flows to the next segment and brings it to threshold. Voltage-gated sodium channels open, regenerating the action potential in this segment of the axon. Current flows from this segment and depolarizes the next segment to threshold, thus regenerating the action potential yet again. In this way, regeneration continues in one direction all the way down to the axon terminals. The basis for unidirectional propagation is revealed when we take a closer look. By the end of the depolarization phase of the action potential, all voltage-gated sodium channels inactivate and voltage-gated potassium channels open. These two events render this segment of the axon temporarily insensitive or refractory to another depolarizing stimulus. However, voltage-gated sodium channels in the downstream segment are closed and receptive to a depolarizing stimulus. Thus, propagation occurs sequentially down the axon to the axon terminals. In myelinated axons, action potential propagation is a bit different. Here they propagate in a saltatory or leaping fashion. The myelin sheath consists of multiple layers of tightly wrapped glial cell membrane. But this sheath is not a continuous one. Exposed areas of axonal membrane, known as nodes of Ranvier, occur at discrete intervals. Voltage-gated sodium channels are abundant in the nodes, but largely absent between nodes. So, action potentials are regenerated at each node, not in areas covered by the myelin sheath. However, the myelin sheath does provide the insulation necessary for the rapid spread of depolarizing current. And the sooner the nodes reach threshold, the faster action potentials propagate along the axon. Saltatory conduction is extremely fast. Velocities often exceed 100 meters per second. In contrast, continuous conduction is fairly slow. Velocities rarely exceed 2 meters per second. Nevertheless, both continuous and saltatory conduction propagate action potentials over varying distances because action potentials regenerate along the way. Summary: propagation of an action potential. Once generated, the action potential propagates the entire length of the axon without decrement.
Okay, so conduction velocity is determined by really two variables, one of which we just discussed, which is number 2 here, the degree of myelin sheath, or is there myelin sheath or the complete lack thereof, because we know that, we either will have continuous conduction or saltatory conduction. But the other variable is axon diameter, and the larger the diameter, of the axon, the faster it transmits. So larger axons transmit faster. The degree of myelinization, of course, increases nerve conduction velocity or transmission. Impulses jump from node to node of Ranvier, called the saltatory conduction. No myelin is much slower. That's the continuous conduction. And I think the video gives you a really good picture of that. All right. Again, the fate of neurotransmitters, we can have diffusion of neurotransmitter away from the synapse and away from where we are have an abundance of receptors for that particular neurotransmitter. Enzyme degradation by, an enzyme like acetylcholinesterase, reuptake of the neurotransmitter or portions of it, into the presynaptic terminal. So those are generally the fates of the three, or the 3 possible fates of neurotransmitters. Now that takes us to the end of the video. And I was just looking at the time. I've been lecturing for about 1 hour and 48 minutes, kind of nonstop. So I apologize sometimes when I get a little tongue-tied. But I will say I am going to try to work on that video. I just passed it now. The video, sorry if I'm getting you dizzy, that did this one. If I can get it to work, it will be working by the time you see this video. So if this will be a mute point. But if I can't get it to work, I'm just going to delete it. It wasn't that great. I think the other ones are much better. I will maybe try to find something else about this and I'll put it in there. But if you're going through the notes and you're seeing this slide and you go, wow, it's not in my copy, it means I just deleted it out. And I didn't want to resume this for almost 2 hours, but I will try to find something that will be a reasonable substitution. Okay, so that's the end of the notes. Sorry about any confusion.